So I said I was going to spend some time on genetics and my sort of favourite area, but let's just look at the pace of change in biology. So you could have added back here Robert Bakewell and the Dishley Lester, and I always sort of, whenever I'm in the RASE building over there, I always pay homage to Robert Bakewell's chair, you know, the founder of breeding the way that we think of it these days. We had 20s and 30s, we had quantitative genetics come along. We didn't understand the structure of DNA until 53. We couldn't actually sequence it in any sensible manner until Fred Sanger worked that out in 1977. Some of the technologies and ideas that were developed by the quantitative people couldn't actually be applied until we had computing power that could do it. And that came best linear unbiased predictors. Those of you who are in breeding technology, that came along because of computing power made it possible. And then we started to get really very accurate breeding values. We didn't have the first molecular genetic diagnostic until 91. And that was the halothane gene in pigs. It's the stress gene in pigs. And now that's completely managed in populations. There are some populations where you want to keep that gene present because it improves meat yield. But you manage the frequency of that gene. And then 2003, the first sequence of the human genome, the first reading of the Book of Life. 5 years, 5 major labs around the world, hundreds of millions of dollars. I've seen one estimate that said $3 billion. The Sanger Centre at Cambridge did 1/5 of the work in one lab, the biggest single contribution of all of them. And then 2004, we had the chicken genome, paid for by the US government as a medical model, not that it was anything to do with agriculture, but as a medical model. Then we started to get lots and lots of DNA markers called SNPs, single changes in the letter code of the DNA, single nucleotide polymorphisms, and most of them don't do anything, but because they're in the population, you can track them. Right, so they're genetic markers that allow you to follow the inheritance of this chunk of DNA in the population from parents to grand offspring and great-great-grand offspring and so on. And it started getting cheaper. And we just had the pig genome published 2 or 3 weeks ago. We've had it for ages, but the paper got published 2 or 3 weeks ago. You can now get a Book of Life read, a pig genome, a cow genome, roughly the same size as a human genome, chicken smaller, for less than $5,000. We're heading towards $1,000 genome sequence. If you spend over $100,000 progeny testing a dairy bull, then it's a trivial expense to also have its genome sequenced. So you can see why in some circumstances it adds up if you can use the information. So DNA is now cheap. DNA sequence is now cheap. And it gets getting cheaper, and the pace of change keeps accelerating. So let's go back a bit and look at the last, 40-ish years, and look at some historical innovations. The biggest one, perhaps, is the change in genetics, breed substitution, and within breed improvement. Texels, Limousin's, the most populous terminal sire breeds. Texels weren't even in the country till 74. And that change has happened relatively quickly. Nothing like as quickly as... some of you will know, what's the fastest growing agricultural technology ever in terms of market penetration? GM crops. Not in Europe, right? But in terms of market penetration, the rate of uptake of a novel technology, GM crops, beats everything in agriculture. Then we had a whole load of innovations that were improving our ability to manage reproduction. Estrus synchronization, induced parturition, multiple ovulation embryo transfer, sex semen, pregnancy scanning, somatic cell nuclear transfer cloning, the Dolly technology. We've had a whole load of growth modifiers, and don't think that this is a dead industry. There is significant research still going on around the world in growth modifiers. It's just that we don't use them in Europe. Everything on that list is legally used somewhere in the world, and effectively and profitably used, and all of them also reduce the carbon footprint of the product. But we don't allow ourselves to use those technologies for no good reason, in my opinion. Animal health, lots of changes in terms of specific pathogen-free breeding populations, so you're not spreading disease down the breeding pyramid. Better diagnostics, better vaccines. You know, who would have predicted that one of the best ways of controlling salmonella in humans via the egg transmission route would be a vaccine for the hen? But it works really well. And disease resistance, and don't think that doesn't apply in my industry. When was the last time you saw a case of scrapie? Right? Single gene, simple biology, or relatively simple biology, and you've already done it. You might have done it because government told you to, right? But you've done it. You have selected for scrapie resistance, and it's almost a disease of history in our populations. Nutrition. Formulation for requirements, improved forages. I'm going to touch on that a little bit more. At least I'll try to. And then a whole load of things for the more intensive or the housed animals in terms of in-feed, synthetic amino acids, enzymes, you name it. I saw a talk from a nutritionist the other day who sort of slightly scared me. We were talking about soya in pig and poultry diets, and his response was, well, the next most affordable protein source or amino acid source is actually synthetic amino acids. And if the price of soya goes up much more, or the price of synthetic amino acids falls a little bit, we'll be using less soya and more synthetic amino acids. That just tells you that other protein sources are not at all competitive for pig and poultry diets. And clever things like immune modifiers. Some really clever stuff going on in the US about changing immune function via things in the diet. Other biotechnologies, GM, agricultural traits, I'll come back to those. But we have the first human pharmaceutical product on the market that's made in a GM animal. It's made in GM goats, and it's a clot-busting drug. And it's commercially successful. And we have a vaccine to control taint in entire male pigs. And then there's a whole load of things gone on post-Farmgate, controlling meat eating quality, much better at understanding what matters post-mortem. If it leaves the farm in good condition from you, it can be messed up substantially in the abattoir, but if it leaves the farm already tough, there's not that much the abattoir can do to put it right. So you've got to get the whole supply chain right. Hazard analysis and critical control points. You've already heard how good we are and how much we're seen as an example by many people in the world of our control of the chill chain. The way that we manage risk and control risk in ready meals and cooked meats and whatever else it might be. We're actually really very good at that. And then traceability and authenticity systems, we're not so good at that. Maple Leaf Foods in Canada can take a DNA sample from a piece of pig meat that a consumer's got at home and can tell you the farm it came from. Because they use DNA, including the mitochondrial DNA, the little power cell DNA in each of the cells. And of course, only the mum has that, and the mum's only present on one farm, whereas the sire might be represented on hundreds or thousands of farms via AI. They can trace a piece of pig meat back to the farm it came from with DNA. And we, in principle, can, just nobody's invested in it. There are obvious limitations on which of those apply in extensive beef and sheep. But not that many beef animals spend no time indoors. And the big one, of course, the one that you can absolutely use, but mostly don't, is genetics. I might be being rude to this audience to say mostly don't, but you'll see what I mean in a little while. Just before I pop back to genetics of the animals, genetics of grasses, I'm not an expert on this, but I guess some of you know the story of the high sugar grasses from Aberystwyth University, commercialised by British seed houses. Because they improve the delivery of energy and protein at the same time in the rumen, they improve the capture of that protein, and less protein gets used as an energy source whilst the flora of the rumen are breaking down the complex carbohydrates. So you get improvements in intake, you get improvements in daily live weight gain in cattle and in sheep, improved carrying capacity potentially. So if you're reseeding, why are you not making sure you're reseeding with the best possible genetics? And there's a win-win. 20% lower methane output. You become, inverted commas, greener. Old slide makes a point. If you're reading the left-hand column, you'll have noticed it doesn't have beef and sheep on it. Because the story is nothing like as impressive. And partly because the data's not so readily available either. But you know, you've heard this morning already this the fact that the New Zealand industry produces just as much lamb from a hell of a lot fewer ewes than it used to. Now, so that actually would be good enough to fit on that sort of table. But a doubling of the kilos of lean pig meat per tonne of feed, an 80% increase in eggs per tonne of feed, and even a 67% increase in milk per cow per lactation. We've not done so well in beef and sheep. Now, there are some really good reasons why you wouldn't do so well. You wouldn't do so well because the generation intervals are longer. Right? You wouldn't do so well because it's more difficult to manage the environment. You wouldn't do so well because each male has fewer progeny, so you can't afford to invest as much in the breeding stock as you would if you were using AI in pigs or something like that. So there are lots of good biological reasons why we ain't so good. But don't lose sight of the fact that animal breeding is an incredibly powerful tool, and you can use it to your benefit. No prizes. Those chickens are the same age. No prizes for guessing which one was selected to put all of its energy and protein into muscle growth, and which line was selected to put all of its energy and protein into egg output. Right? Really powerful tool. The world's broiler and layer industries are dominated by a handful of companies. 3-ish. Right? One with lots of breeding stock up in Scotland has a 50% global market share of broiler genetics. Compare that to what we do. But actually, what we've done to livestock's nothing in comparison to what we've done to our best friend. Right? So there's lots and lots of scope to do better. You're terribly familiar with the notion, I don't need to take you through this, that those breeding decisions at the top of that pyramid have quite a big impact down the bottom. And I'll give you the broiler example again. One male at the top of the pyramid, follow his genes down to the slaughter generation, and you'll find his genes in over 60,000 tonnes of poultry meat. Quite a responsibility making those breeding decisions. And the companies will sink or swim by those breeding decisions. And we looked at a report from DEFRA a few years ago where they'd paid Cranfield University to build some really quite neat life cycle analysis models of UK agriculture, and asked the question, hasn't anybody put genetic improvement in there? And no was the answer. So DEFRA paid us and the folks at Cranfield to do this. And we were lucky because the breeding organisations trust us to give us their genetic trends without publishing them, which we then amalgamate and then put those figures into this Cranfield model. So you've got methane, ammonia, nitrous oxide. Obviously, the methane and nitrous oxide much more potent greenhouse gases than just CO2. And you can multiply those lot up and you get the global warming potential for 100 years. So you've got 20 years genetic improvement and the 100-year global warming potential in the red box at the right. Layers and broilers have knocked more than 1% a year off their carbon footprint, if you like, off their global warming potential, just through breeding. Not so good when you get down to beef and sheep. However, just for the hell of it, we said, but there has been genetic improvement in sheep. It's just not widely taken up. Put the genetic trends for the performance recording sheep breeders into this model. Just the performance recording sheep breeders. And that -1, if you swapped every ram in the country for an average ram from a performance recording pedigree breeder, that -1 would be -8. Think about it. Wonder how long it'll be before retailers start asking why you're not doing it. And it's a win-win, for heaven's sake. You know, you get more productivity, you get better carcass quality. It's a win-win.